The macula is the sensory organ for both the saccule and the utricle. It's exactly the same sensory organ, but there's one small change between the saccule and the utricle. Okay, like I said, the utricle is responsible for detecting linear motion, whether it's forward, back, side to side, and that is because the macula is on the floor of the pool with the hair cells sticking up and the weight causes cilia motion that way. So if I told you that the saccule was responsible for detecting up and down motion, tell me, where is the macula located in the saccule? On the side of the pool, yes. Everybody see that? Okay, cool. Let's see if I can draw it. Where's my green? There it is. Okay, so first we have to draw the hair cells. So here I am on the side of the pool. Okay, so I'm going to draw my hair cells, and they're going to be coming down here. So this is on the side of the pool. Everybody see where I'm? Okay. So this is going to be the hair cells on the side of the pool. On the side of the saccule. Okay. And just like before, we have the axons coming out of the bottom of the hair cells. Okay. These axons are going to come out of the bottom of the hair cells, every hair cell, and it's going to connect up as part of the vestibular portion for the 8th cranial nerve. So all these axons coming out are going to be part of that 8th cranial nerve. And it's going to be the job of the saccule to detect that up and down motion. So this is the thing that's going to be active when you get in an elevator and you know you're moving up or down. Okay? So these are nothing more than the axons coming out of the bottom of the hair cells of the saccule, and they're connecting up, and they come out one side and the other as part of the vestibular portion of the 8th cranial nerve. Okay, coming off the top of the hair cell, top of the hair cells, just like we had before, are going to be your cilia. So these guys are just pointing straight out. Just your cilia pointing straight out, and then over top of that we have our gelatinous matrix. So here's the gelatinous matrix in 3 dimensions, and that's just overlying those cilia and hair cells. And then adding weight to the entire system to detect, gravity helps it detect whether you're up or down, going up or down, are going to be, here are your otoconia, just nothing more than those calcium carbonate crystals, same as the other one. Okay, and realize, somebody asked the question, I think it was Ben asked the question, is there that endolin fluid? The endolin fluid is all inside of here. Okay, so these two are connected. So all this to the cochlea, that endolymph is all throughout both the saccule and the utricle, and these are on the way to the semicircular canals. It's all endolymph filled, so it's filled with a fluid known as endolymph. Okay, so can you see? Well, maybe you can't see, but I can tell you that if an otoconia becomes dislodged from the macula sensory organ and is free floating in that endolymph, it can get lodged into the semicircular canals since it's all connected. It's all a connected system. Do you know what it's called when it gets lodged into the semicircular canal? Yeah, BPPV, benign paroxysmal proximal vertigo. Yeah, it's called BPPV. We're going to look at a video on BPPV. But that's exactly what happens. BPPV. You have a question?
Can you write it on the board and like describe?
BPP, yeah. We're going to watch a video. We'll talk about it more later. It's called BPPV. BPPV. So, benign paroxysmal, it's called benign paroxysmal positional vertigo. Okay, BPPV. In BPPV, when I talk to patients, I say nothing more. The rocks in your head got knocked loose, and I got to put them back in place. Okay, and that will be your job. You'll learn how to treat BPPV next year. And then that's all it is. A lot of times people will wake up in the morning, sit up in bed, and imagine one of those otoconia got knocked loose, and it just gets stuck right into the organ that it's connected to, and it causes vertigo. Okay, we'll look at how that happens here. It's a common problem. Questions about? Your dad had it? I've had BPPV. It's no fun. I would have thought I was having a stroke if I didn't know better. Luckily, I knew better. Questions? Yes.
So, are there different types of vertigo? Like people who just have, or is it always caused by an otoconia? That's the nerve.
Vertigo is a subjective complaint of the world spinning around you. Very specifically, that's what that means. The most common cause of vertigo is BPPV. However, you can have dizziness and/or vertigo associated with any problem with the vestibular system. It could also come on due to a cerebellar stroke, brainstem stroke, anything with the whole vestibular system, the central pathways. We'll talk more about that. It's called vestibular hypofunction. And you'll learn how to treat that next year too. Common after concussion. Any other questions? Okay, moving on. This is the saccule and utricle sensory organs. Now, we're going to look at the semicircular canal sensory organs. Okay, put that up here. Here's the best way I can say it. So, you see how this, so going back to this picture, all I did is I took off the lateral canals for both the right and left side. Okay, so I'm taking this lateral canal from the right side and I put it here, and then this lateral canal from the other ear, the left side, and I put it over here. Okay, to make that easier to see, I'll go ahead and separate these. So this is the right side and this is the left side. And here's my midline. Okay, and here, this is the opening or the hole that's leading to the vestibule. Okay, so let's go ahead and label that. This is my right horizontal or lateral, whatever you want to say. Okay, and then this is my left horizontal semicircular canal. And this big bulbous part of the bone, this is known as your ampulla. Okay, so there's an ampulla on the right side and an ampulla on the left side. Okay, if you remember, coming off the ampulla, this opening here is to the right vestibule, and then this opening would be to the left vestibule. What's happening? So all I've done is I've taken the right horizontal semicircular canal and I've just pulled it out, and I've taken the left horizontal semicircular canal and pulled it out of the vestibular, and I'm just looking at this in isolation. Just two different canals. I like to look at the horizontal canals. Horizontal canals are responsible for detecting head shaking motion of no. Okay, so they're horizontal, so as you move your head, it detects that motion, that no head shaking motion. We're going to see how it works. Okay, like I said, the macula is the main sensory organ for both the saccule and the utricle. It contains otoconia, and the main sensory organ for the horizontal semicircular canals, all three of them bilaterally have their main sensory organ in this area known as the ampulla. Okay, and just like I had shown previously, lining the semicircular canals, there's this membranous labyrinth. Let's go ahead and draw that in here. So here's your membranous labyrinth. Okay, that membranous labyrinth is nothing more, it's just lining the bony labyrinth portion of the semicircular canal on both left and right sides. And that is your membranous labyrinth. Membranous labyrinth. Okay, nothing more, just a quick review of what we've seen previously. The new part is inside the ampulla, like I said, is going to be your sensory organ for motion detection, head motion detection. And similar to the ones we just looked at, to the macula, it's also composed of hair cells. So I'm just going to draw my hair cells here. These are my hair cells. Whoa. And just like we saw previously, out of the bottom of the hair cells, it's going to be axons that connect up on the left side, and axons that connect up out of the bottom of the hair cells on the right side. And both of these are going to be part of the vestibular cranial nerve. So this is going to be the right portion of cranial nerve, or cranial nerve 8, the right vestibular portion. And this is going to be the left. Okay, so these axons are just coming out of the hair cells. They're part of the main sensory organ that resides in the ampulla of each of the semicircular canals. These axons all come together and connect up with the axons coming out of the saccule and the utricle as well on each side, and help form the vestibular portion of the eighth cranial nerve. Okay, now just like the macula, there's going to be some cilia involved, but they're a little bit different. Okay, the hair cell that's closest to the vestibule opening. Okay, so this hair cell has a great big old cilia. The other cilia are kind of short. Okay, so on the right side, the cilia furthest, or the hair cell furthest to the left, is closest to the vestibule opening, and that's going to be a great big old cilia. The great big old cilia has a special name. It's known as the kinocilia. K-I-N-O. The kinocilia. Okay, if you want to think about it as king cilia, that's how I remember it. Kinocilia or king cilia, because it's great big old cilia. And the same thing happens, that's on the right side. So on the left side, it's the hair cell that's furthest to the right. It's closest to the opening to the vestibule. That's where the great big old cilia resides. The other cilia are kind of short. Okay, and that great big old cilia is known as the kinocilia. Okay, there's also going to be a gelatinous matrix involved. I like to think of this gelatinous matrix as looking like a cup. So that let's think about an upside down cup that encompasses all of these cilia, as well as the tops of all the hair cells. Okay, so on both sides there's going to be this cup-like structure that encompasses all the hair cells and all the cilia, And I like to think about it as a cup because the name of that gelatinous matrix is a cupula, C-U-P-U-L-A. So C-U-P-U-L-A is nothing more than gelatinous matrix. Okay? And this gelatinous matrix, known as the cupula, encompasses all the hair cells and all the cilia that are coming out of the top of the hair cells. Okay, there's one big difference between this sensory organ and the macula. There's no otoconia. Semicircular canals have no otoconia associated with it. All we have are the hair cells, the cilia, with the kinocilia being closest to the vestibule, and the cupula. It doesn't have the otoconia. It doesn't have those heavy calcium carbonate crystals that the utricle and the saccule have. Okay. No otoconia in this system. Yes, Susan?
Did the gelatinous matrix and the saccule and utricle on the macula, did that one have a name?
Nope, no specific name. Basic jello. This one's called the cupula. Any other one, Ben?
So do you see any depolarization in this one? You don't have any of the weight on the hairs?
Yep, I'll show you that in a minute. Very good. Good prompt. Very good lead. Any other questions? Okay. There's one more thing that I didn't. I told you about previously, but I haven't talked about before, and that is this system. This whole semicircular canal is filled with a fluid, right? And remember the name of the fluid? Endolymph, yes. So it's filled with a fluid known as endolymph. So endolymph's just that fluid. So Ben's question is there any depolarization associated with the hair cells? And it all has to do with the fluid and motion. And the best way I know how to say this is to show you. Okay? So let's imagine that this person or this person is going to turn their head to the right. Okay? They're going to head turn to the right. Let's make it easy to see. So in other words, here's my horizontal semicircular canals. If it were me, I'd be turning my head to the right. Let's look to see what happens. Okay. I know you guys have one of these. Let's see if I can do it. You guys, this is called an inclinometer. Have you used it yet?
Yep.
Oh, really? Okay, I didn't think you'd used it yet. So that's helpful, actually. It's for spine. Oh, you've done spine already? Okay, good. You've done lumbar. Awesome. Let's see if I can put this up. So what has to happen is it has to. Well, if you have one, you can play with it. Can everybody see the? Here, let me see if I can do it this way. You're going to see you, but that's okay. There we go. There we go. Okay. Let's see if I can set it up so that it works. You still see that? There we go. So I'm sort of zeroed out over here, right? Okay. And Paul's trying to put his hand in to get. Okay. Now I'm turning my head to the right. So if I do that, I do that. Which way does the fluid move? Can you see that? To the left, right? So if here's my 0, and if I turn my head to the right, can you see how the fluid goes that way? Right? So that's the key. The key is if I turn my head to the right, the fluid moves left. If I turn my head to the left, the fluid moves right. Okay? And if you get your inclinometer, you can prove that to yourself just to watch it happen. But that's going to be the key to the whole thing. So in answer to Ben's question, when this person turns their head to the right, the endolymph is going to move this way. Right? In the right horizontal canal, if they turn their head to the right, the fluid's going to move to the left. Everybody see that? So every time the fluid moves, every time this kinocilia bends toward the vestibule, it causes depolarization. Okay? So if I'm turning my head to the right and the kinocilia bends toward the vestibule, that causes depolarization. That's excitatory. Let's put it in green. Green for go. So this side is going to be depolarized or excitatory. Does everybody see that? Okay, that's happening. Go ahead.
Move up a little bit. I can't see it.
Oh. Okay. Yes, this side. Signals coming down this portion of the vestibular cranial nerve number 8 are going to be excitatory because the endolymph is moving that way, which causes the kinocilia to bend toward the vestibule. And this is endolymph. Okay, so signals coming from my right horizontal semicircular canal are going to be excitatory. I'm going to get depolarization. Okay. Now, if I move over to the left ear, looking at the left ear. So I'll turn my head to the right, fluid goes to the left, so the fluid's endolymph is flowing this way. In this case, the kinocilia is bending away from the vestibule, right? So if it's going this way, it's going bending away from the vestibule. This is going to be inhibitory. Okay, so I have an excitatory signal coming from the right side, and I have an inhibitory signal coming from the left side. Does everybody see that? Questions about that? This is going to be important for moving forward. Yes, Ben.
So it's the pressure from the endolymph fluid on the I forget what it's
Cupula.
called.
Cupula.
That's causing the depolarization.
So yeah, and so I haven't named the sensory organ. I don't think I have yet, but I'm going to give you a name for that whole sensory organ. But it's that pressure. It's like you imagine water flowing through a garden hose. It goes whoosh, right? That's going to cause bending toward the vestibule, excitatory. Bending away from the vestibule on the other ear is going to be inhibitory. Okay. Any other questions? Need you to kind of get the concept to see where we're going here. All right. The only thing I haven't done, I gave you a name for that main sensory organ in the saccule and the utricle. There's a name for this whole sensory organ here. So this whole sensory organ. Hair cells, kinocilia, and the cupula is called your crista ampularis. So it's C-R-I-S-T-A, ampularis, because it's in the ampulla. Your crista ampularis. The crista ampularis is just the name of the main sensory organ for each of the semicircular canals. Write that down. I'll just put canal sensory organ. And again, the big difference between the canal, semicircular canal, crista ampularis, and the macula is going to be that macula have otoconia. Crista ampularis has no otoconia. It just depends on the position of the bending of that kinocilia in response to head movements. Questions? You don't understand that. Johnny, you're looking? Not to answer a question, or?
I'm not confused. I'm just squinting.
You're squinting, okay.
Yeah.
Is there something you can't read? Is there something unclear?
I was reading what you wrote under Krista.
Okay. It's a canal sensory organ. Yeah, it's the main sensory organ for the canal. I kind of ran out of space, so I showed it a little bit. But both of those are known as the crista ampularis. And pretty basic system. No questions? Let's move on to the vestibular system, central pathways. Okay, back to something that looks a little bit more familiar, I hope. Yeah, okay, so this is going to be my standard frontal section. Oh, sorry, Gabby, question? Sorry, can you speak up a little? That's not dependent on otoconia at all. It's only dependent on the head motion causes the pendulum to flow fast in the opposite direction, which causes that kinocilium to bend either toward or away from the vestibule. What is that dependent on? Forward or linear motion. Motion forward or back. So for the utricle, you're going to detect motion. So if you're sitting in a car and that car moves forward, you know how you can feel that you move forward even though you're not doing the moving, it's the car that's moving? Okay, that's going to be your utricle. Your saccule is going to be most involved when you go up and down in an elevator. You know how you can tell if you're going up or down? That's your saccule. And that's dependent on the position of that maculosensory organ. The utricle is on the bottom of the pool, and the saccule is on the side of the pool. So it's going to be the weight of the otoconia that is either stimulated by motion, or inertia, and gravity. Does that make sense? Yeah. Any other questions? Okay, moving on. This is going to be this is vestibular system central pathways, and hopefully my standard frontal section. So here, you see that? Here's the cortical region. Here's my thalamus, midbrain, pons, medulla. Back is the cerebellum. Okay. Hopefully this looks super familiar to everybody. And what I like to start out by doing is just drawing in the main players in the system. And I can tell you, we covered this previously, so this is going to be a little bit of the review when we talked about the cerebellar region. We talked about this nucleus here that was involved. That's going to be your vestibular nucleus. Okay, I'll go ahead and draw it in bilaterally. So here at the caudal pons, you have your vestibular nucleus. Okay, and coming out of axons coming out of the vestibular nucleus stimulate this tract that I like to use so much clinically. It's your vestibulospinal tract. Review to way back. Does anybody remember that portion of the cerebellum that's associated with the vestibular nucleus? So the functional anatomy region is the flocculonodular lobe, but it also is known as the vestibulocerebellum. So the functional region is the vestibulocerebellum. The gross anatomy is the flocculonodular lobe. It's going to be here. Okay. Let's talk about functional regions. This is going to be your vestibulocerebellum. There's also associated with the vestibulocerebellum, there was a deep cerebellar nucleus. Does anybody remember the name of that one? Good, this is your fastigial nucleus. Let's bring back some memories. And then sitting in here in the vestibulocerebellum, there's going to be this cell type, this Purkinje cell that's here. It sends out axons that are associated with this vestigial nucleus. And here to allow passage of the axons from the pons to the cerebellum and back again is going to be your inferior cerebellar peduncle. what we talked a little bit about today, where the cranial nerve number 8 axons came from. So we talked about the vestibular portion of cranial nerve number 8, right? And that's coming from, those are nothing but axons coming out of hair cells from the macula, the utricle, and each of the semicircular canal sensory organs. So that vestibular portion of cranial nerve number 8, now we can see, comes in and synapses with this nucleus, which is the vestibular nucleus. Axons from the vestibular nucleus. As a review, we showed them before, coming out of the vestibular nucleus, coming into the cerebellum through that inferior cerebellar peduncle, and then synapsing directly here with the Purkinje cell in the vestibular cerebellum. And that Purkinje cell is hooked up already to the vestigial nucleus. Vestigial nucleus sends out axons that come down and go back out through the inferior cerebellar peduncle and synapse with the vestibular nucleus. Okay, this is how the cerebellum knows where the head is in three-dimensional space at all times. So far it's a review. We have a couple things more to label. Let's make it this blue color. Okay, now we're going to link this vestibular system up to the cranial nerve nuclei that are involved in extraocular eye movements. What cranial nerve nuclei is located in the medial pons that has to do with extraocular eye motion?
Abducens?
Yes, abducens. So here's cranial nerve nucleus number 6. Here's abducens. Up on one side and on the other. To make it clear, I'm going to go ahead and put my midline in. Didn't do that yet. So you can see that I'm doing this bilaterally. Okay. So this is abducens, cranial nerve number 6. Right? Located most medially in the caudal pons, abducens nucleus. Okay. Then moving on up, we saw another cranial nerve nucleus that's going to be located here at the level of the inferior colliculus. Does anybody remember the name of that? Which cranial nerve nucleus is located in the midbrain at the level of the inferior colliculus? Trochlear, cranial nerve number 4. Right, you're close. Okay, that's going to be here in the middle. So at the level of the inferior colliculus, we have your trochlear nucleus, so cranial nerve number 4. Remember? And then at the level of the superior colliculus, we have cranial nerve number 3. So this is, let me label that. This is cranial nerve number 4, so that's your trochlear. And then just about that, we have cranial nerve number 3. Cranial nerve 3 is in the midbrain at the level of the superior colliculus. Okay, this is cranial nerve number 3. And these are all involved in extraocular eye muscle innervation. Okay, so 3, 4, and 6. So just connecting two things that we've learned previously. Now we're just going to put them all together. Everybody got it? Questions about what we have here? So all I'm doing is, this is the portion of the cerebellar region that we talked about, vestibulocerebellum, where the vestibular nucleus is hooked up to the vestigial nucleus in the cerebellar region, comes back out and signals. Today we talked about the vestibular portion of cranial nerve number 8, where exactly that comes from. These are all axons coming in to give information to that nucleus, to that this vestibular nucleus. Now, we also talked about cranial nerves involved in extraocular eye motions, 3, 4, and 6. So 3, most of the muscles, cranial nerve 4, superior oblique, cranial nerve 6, lateral rectus. So what you see, and I'm just going to draw this in I'm just going to draw this on one side, because if I did both sides, it'd be complicated. So let's consider the right side. So coming out of the right nucleus, there are axons connected to the ipsilateral as well as the contralateral cranial nerve 6. There are also axons coming out of this right vestibular nucleus that are connected with cranial nerve 4 ipsilaterally as well as cranial nerve 4 contralaterally. And then finally, there are axons coming out of this vestibular nucleus on the right side connected to cranial nerve number 3, nucleus oculomotor, ipsilaterally as well as contralaterally.
See those here? Where are they coming from?
So these axons are coming out of the vestibular nucleus on the right side, and they're connected with both the right and the left cranial nerve 6, cranial nerve 4, and cranial nerve 3. Now the same situation happens on the left side with this vestibular nucleus, but I'm not drawing it in because it's too complicated. Right? I'm just drawing in one side. But realize all these connections are made bilaterally. That'll be important here in a minute. It's also important to note that this vestibular nucleus sends out an axon that comes all the way up and synapses with the contralateral nucleus in the thalamus. Okay, synapses, this is just a nucleus, any old generic nucleus in the thalamus. We don't have to know what kind or which one. And then this nucleus sends out information to the somatosensory cortex. It's a complicated system. I thought Johnny first.
So the ones that go to the thalamus are also coming out of the vestibular nucleus?
Yes. Is that your question, Caitlin?
Kind of all of those are coming from the vestibular, so those are all the same orange color.
Yes, they're all the same orange color. They're all coming out of the vestibular nucleus. In this case, I've chosen to represent it only coming out of the right vestibular nucleus, but I realize they're all coming out of the left as well and hooking up bilaterally. So all those connections always exist there. And that's going to be important here in a minute. Any other questions about what I'm trying to show? And that's the whole point here, is that the information from the information coming in through the vestibular system is intimately connected to eye movement. We're going to look at that in a second. And this is actually something that I talked about when I did the cross sections of the brain stem. These form a tract. I'm not going to include the vestibular tract. And this tract that we looked at previously, does anybody remember what I talked about for coordinating eye movements? Your medial longitudinal fasciculus. So this forms the tract. So your medial longitudinal fasciculus is nothing more than axons coming out of the vestibular nucleus bilaterally that connect the vestibular nucleus to the different cranial nerve nuclei involved in extraocular eye motions. So this is your medial or your MLF bilaterally. Okay.
What did you say again?
Medial longitudinal fasciculus. And when you do a cross section of the medulla, it's kind of hanging around with the medial meniscus. Medial, like right on top. It's a little black. I've represented it as kind of a medium-sized black dot. Okay, and that's what this is. That's what you're looking at. Questions? Okay, let's look at the next one. Let's look at something called vestibulo-ocular reflex. An acronym would be VOR. Okay, this is what VOR is. I'll write that up here. Vestibular ocular reflex, or VOR. Okay, what you see, this is the right side. This is the left side. These are eyes. This is your right eye. This is your left eye. And these are your semicircular horizontal canals. Okay, one on the right and one on the left. This is just a frontal section through the brainstem, midbrain, pons, and medulla. Okay, everybody got it? A lot of information here, so we'll just take it one step at a time. What I like to do is put in all the players, and also here's your midline. So going through and looking at the brainstem, I always like to put in my midbrain. And I talked previously about 3 different nuclei. Let's look at what colors I use so I can keep it consistent. 3 different nuclei that are involved in extraocular eye motions. And down here in the medial pons, does anybody remember what this one is? Abducens. Great. For cranial nerve number 6, it's going to be abducens. Okay. I also talked about this nucleus that's involved in receiving vestibular information. I'm going to show you how he works today. Okay, that's going to be your vestibular nucleus on the right side and on the left side. And moving on up in brown, I also talked about the 4th cranial nerve, the trochlear nerve nucleus here. It's going to be at the level of the inferior colliculus in the midbrain. Okay, the last one I used was this color here. This is going to be your ocular motor nucleus. Present bilaterally, so cranial nerve number 3. So that's your oculomotor nucleus. Okay. So nothing new. I also talked about in this horizontal, semicircular horizontal canals, they are lined with this membranous labyrinth here and filled with this endolymph fluid. There's my membranous labyrinth. About the right and the left sides. And their method for detecting head movement is going to be that they have hair cells that come up that reside in this ampulla region. Okay, hair cells coming up bilaterally. And those hair cells have closest to the ampulla, they have a kinocilia and then a couple of small cilia. The one closest to the ampulla is the kinocilia. Nothing more than what we just did, and overlying the cilia is going to be that cupula, that gelatinous matrix known as the cupula. All right. Okay, and coming out of the bottom of the hair cells, we have the vestibular portion of the 8th cranial nerve. They're all coming in here together and then synapsing with the vestibular nucleus on the right side and then on the left side. everybody set up and ready to go. We are going to do a simple experiment, and during the exam, I do not care if you do the experiment. You can do it as many times as you want. Okay, this is how I remember it. On the right, what we're going to do is we're going to have this person turn their head to the right. Okay, again, we're going to right head turn. If we have a right head turn, if everybody looks straight ahead, okay, with their eyes, have your eyes focused straight ahead on something. And what I want you to do is keep your eyes focused straight ahead, but turn your head to the right. But keep looking straight ahead. You have to keep your eyes forward. Okay? Now tell me, your left eye, does it move medial or lateral? Lateral. Which muscle does it use in your left eye to move laterally? Lateral rectus, abducens. Great. So let's go ahead and green for go. We're going to put over here, when we turn our head to the right, I'm going to have an active lateral rectus. Okay. Again, if we do that again, we turn our head to the right. Which way does the right eye move? Medially. What muscle does it use to do that? Medial rectus. So over here on the right side, I'm going to have an active medial rectus. Okay, the medial rectus is active. Now if the lateral rectus is active on the left side, inactive on the left side is going to be its counterpart. It's going to be its medial rectus on the left is going to be inactive. All right? Does that make sense to everybody? So if the lateral rectus is active, the medial rectus has to be shut down. It's the antagonist. So medial rectus on the left is inactive. And same situation on the right. If the medial rectus is active on the right, then I want the lateral rectus to be inactive. Okay? Everybody got it? Understand what I'm doing? Does that make sense to everybody? Just looking at what muscles are active and what are inactive, and based on that, if you know that if you turn your head to the right and the endolymph is going to flow this way, the kinocilia is going to bend toward the vestibule. So information coming from this vestibular nucleus is going to be excitatory. I'll make it in green for go. This side is going to be excitatory. And same way, you turn your head to the right, kinocilia bends away from the vestibule. Information coming in from this horizontal semicircular canal being relayed to this vestibular nucleus is inhibitory. I'll put that in red. Okay, this is just going back to what we talked about previously. Does everybody remember talking about that? Got it? So one side is going to be excitatory and the other side is going to be inhibitory. So all we have to do is look up here and figure out what gets excited. Okay? Now I know that the medial rectus here on the right side is going to be active. So that means that this side that's excitatory has to come up here to the right oculomotor nucleus and synapse. And it also has to synapse or depolarize the axon that's synapsing with the left lateral rectus, because those are the ones that are working. Does that make sense? You don't have to memorize anything. If you do the experiment, you can figure it out. And conversely, here from this side, from the left side, this left information coming in is inhibitory. So what's inhibited On the left side, I want to inhibit my medial rectus, which is innervated by my cranial nerve number 3. So that's going to synapse and be inhibitory. And it's also going to synapse here. It's going to also inhibit cranial nerve 6 on the right side because that's the one that I want to be inactive. So I send out a signal here. Synapses is inhibited. And that's the VOR, vestibulo-ocular reflex. It's going to be one of those tests that you learn when you look at vestibular next year in our rehab in the spring. Questions? Good? Okay, let's take a 10-minute break.